AQA Biology, Lipids. As I begin all of my videos, we're going to go over the key terms for this unit. Triglycerides are made up of 3 fatty acid chains attached to a glycerol molecule. Fatty acid chains attach to the glucose molecule in a condensation reaction. 3 molecules of water are formed, and the bond holding the two together is called an ester linkage. A saturated fatty acid chain contains no double bonds between their carbon atoms. These fatty acid chains are saturated with hydrogen, so they have the maximum amount of hydrogen present in the molecule. Unsaturated fatty acids contain a double bond between the carbon and carbon. Now, this causes a kink in the chain, which means that generally unsaturated fats are liquids. Monounsaturated means that there is one double bond present, poly, many, as described in the other videos. And if you don't really know what the abbreviations between the words mean, I really do suggest you looking at the first 2 videos as we go into detail on what they actually mean. Phospholipids contain 1 glycerol, 2 fatty acids, and 1 phosphate molecule. Lipids. Now, some characteristics of lipids is that it contains carbon, hydrogen, and oxygen, and the proportion of oxygen and hydrogen is smaller than that in a carbohydrate. It's insoluble in water and is soluble in organic solvents such as alcohols and acetone. Now, there are 2 main groups of lipids, which are triglycerides and phospholipids. Now, a triglyceride is formed between a reaction between 3 fatty acid chains and 1 glycerol molecule. 3 condensation reactions occur as the OH bond is broken, leaving a joining of the fatty acids with the glycerol. Now, this creates a triglyceride with 3 molecules of water formed as well. This is formed in a condensation reaction and can also be broken in a hydrolysis reaction. And if you don't really know what these are, then really do suggest you looking at the previous videos. I'm pretty sure I'm going with the indication that you are quite solid on what those mean right now. Roles of lipids. The cell membrane is flexible and allows lipid-soluble substances to pass. It's a source of energy. It allows waterproofing, as water can enter and exit the cell. It acts as insulation for the cell and protection for the cell. Structure of triglycerides. It contains a high energy bonds in the carbon hydrogen bond. It has a low mass to energy ratio, which makes a good store of energy in a small volume. It's insoluble and a source of water due to the high ratio of hydrogen to oxygen bonds. Now, the uses of triglycerides. It acts as an energy store, it's an insulator, a protector, and a hormone. Now, this is generally the structure of a phospholipid. As you can see, they have the phosphate group, a glycerol group, and the 2 fatty acid chains. And this is the general structure. So due to this structure, as you can see, one side of the molecule is negative and the other is positive. This creates a polar molecule due to its hydrophilic head and hydrophobic tail. Phosphate loves water, which is hydrophilic, and the fatty acids actually repel water, which is hydrophobic. Now, the phospholipid structure is important as glycoproteins are important in cell recognition. Now, how do we test for lipids? Well, this is one of the required practicals if I can remember correctly. And how you would test for them is you add ethanol plus another acid, and then you shake, add water, and if it goes cloudy, then a lipid is present as an emulsion is formed. Now, this is all there is about lipids. It's quite a small unit, but the next video will be about proteins, and enzyme actions, and as well as inhibitors, as they all kind of fit together quite nicely, I find. Thank you for listening and subscribe.